how is the information in DNA converted into the structure of a protein. In this video, I'll show you DNA structure and transcription of DNA to messenger RNA. Here's a cell with a nucleus. I'll just show a small part of the DNA in that nucleus with some base pairs. So I've shown you two triplets on DNA. And of course there would be more with bases on each side. And all of these base pairs are weakly bound to each other. Any gene here would include all of these triplets and many more on each side. A single gene might have 1,000 or more base pairs or 300 or more triplets. And a chromosome might have 1,000 genes. I want to focus just on this portion of gene X. To remind you, if we zoomed in on that DNA strand, we'd also have sugar, phosphate, and the bases on each of these. Here's our cell again. Here's some neighboring cell nearby. Here's the nucleus. In the nucleus, where we have our DNA strand, our A's and T's paired up, C's and G's, all the way along, with more bases. A protein binds here that copies from one strand of the DNA. So this protein moves along the DNA. Coming out will be a strand of messenger RNA. That strand of messenger RNA, remember, also has a sugar, a different sugar, phosphate, sugar, and again, from the sugars are bases. We replace the T's that were in DNA with U's for uracil. As our protein moves along, remember it's making a strand of messenger RNA here. It's copying from one of these strands. If it's copying from the top strand, it's going to put matching bases on. So the matching base for A would normally be T, but this is messenger RNA, so it's a U. The matching base for C would be a G and G. As our protein moves even further along, it will have also copied from the GTC and will have a CAG. So the strand of DNA was TGGCAG, but the strand of messenger RNA replaces the T with a U, UGGCAG. So the GGCAG are all the same, but the T has been replaced with a U. And the result will be a strand of messenger RNA out in the cytoplasm, and our triplets have now been turned into what are called codons, UGGCAG. Here are the sketches to practice. Cell, nucleus, DNA, A's and T's, weakly bound, C's and G's. One triplet is enough to remember. It's possible to practice the whole story of transcription in a single sketch with each piece connected, but it's easier to remember if you break it into smaller sketches and combine each into the pieces of a story. So in this second sketch, I'm showing you the protein binding to DNA and moving along DNA and transcribing, making a strand of messenger RNA, replacing the T's in DNA with U's for uracil. And in this third sketch, I'm showing the transcribed strand of messenger RNA now in the cytoplasm with the triplets from DNA having been transcribed into codons on messenger RNA.